So we're going to look at different methods humans use to control reproduction for various purposes, and I suppose the hard bit about understanding this is recognising when you've got sexual reproduction happening, when you've got asexual reproduction happening, which bits are cloning and which bits aren't cloning. So these are the methods we're going to look at. You can see the bottom two you don't really specifically need to learn for AQA, but they seem like common sense, so I've included them here just for interest really. So they're the methods we're going to look at, and when we look at each of these 6 diagrams we'll just ask ourselves these questions quickly. You could fill it out as a table if you want. I've made this video pretty quickly. You could do it that way. So we've got, first of all, here a process you need to be able to identify. This is taking cuttings, and it's something that you can only do in plants. And so you need to ask yourself, is sexual reproduction here? No, this is just a sexual reproduction. So which animals are clones? Well, these, well, they're plants anyway, but these 2 plants are clones of each other. They're also clones of the parent plant. And why are they clones? They all contain the same nuclei in their cells, and the nucleus contains the chromosomes, which contains the genes, which is the code for the characteristics. I suppose, where exactly does the asexual reproduction occur? Well, this is something that happens here as soon as you take the cutting from the parent plant. You've got that's when you create more individuals. Of course, this happens naturally anyway. You've got potatoes have potato tubers, strawberries have strawberry runners, but it's something that humans do a lot. Now this is slightly different. This is plant tissue culture, and you can see that this time, instead of taking a whole leaf, you've only taken 1 cell from the parent plant, and it's put into a petri dish with some nutrient, with some agar jelly here, and that cell starts to reproduce, and or to multiply rather, and then you get a plant growing from it. Now that's something we don't do with animals, but plants are able to do that, and animal cells aren't able to do that. So does it create variation? No, there's no sexual reproduction here. Only sexual reproduction creates variation. This plant is a clone of its parent. Why? Well, it's the same answer. Why? They've got the same nuclei in the cells. The nuclei contain the genes and the same chromosomes, and the genes are the code for the characteristics. So this you need to be able to identify as genetic modification or genetic engineering, and you can see if we look at cows in all the next few examples, we've got a cow here, and the only 1 gene from the strawberry is being selected. You can see 1 cell is taken out of the strawberry. We zoom in on the nucleus of that cell, and you can see a pair of chromosomes here, and 1 chromosome is selected, and that chromosome would code for a certain characteristic which is desirable, that we want as humans. Let's say, for example, it's the code for strawberry flavour, so we just take out that chromosome and we move that across into the nucleus of this cell here. So this cell now, the cell of the cow, which can grow into a new cow, now has only 1 specific gene in it, and that gene will put maybe the flavour of strawberries into the new cow. Now there's a tiny amount of DNA coming here from the strawberry. Most of the DNA of this cow obviously comes from this cow, doesn't it? So this is nearly identical to this, other than the fact we've only put 1 gene into it. Sometimes this is called a transgenic organism, because trans means kind of across, I suppose. You've just moved 1 gene across, so transgenic. This is a transgenic organism. So sexual reproduction? No sexual reproduction here. This is more or less identical to that one. other than the one gene. Is there any variation created? Well, yes, this is quite different to that, although it's not variation as we usually think of in biology and in natural selection. There are no clones, and there's no asexual reproduction. So that's genetic modification. Now, these diagrams look, if I just flick back and forth between the next two, they look like a bit of a headache, and you need to be able to recognise the difference between these two. You've got here, and this is fusion cell cloning, sometimes called adult cell cloning, and you've got no sexual reproduction. You've got a clone. You've got the nucleus coming from one parent. Of course, the nucleus contains all the genes and the DNA, the chromosomes from one parent, and they only come from one parent, and they end up in this new organism here, the next generation. This is a clone of that. So these two either end. And I mean, a typical exam question would say, or ask you to identify which animals are clones, and it would ask you to say why are they clones, at which point you launch into your list of using keywords like DNA, chromosomes, characteristics. So it's important to recognise here all that this parent's doing is donating an egg, and there are no characteristics from this parent, so these two share no characteristics. Also, this organism here, known as a surrogate mother, shares no characteristics with any other organism that's shown on the screen. So all that happens is the new cell here grows into an embryo. The embryo is implanted into the surrogate, and all that happens there is that this embryo develops within the surrogate and until it's ready to be born, and then you get your clone. That is a clone of that. Any sexual reproduction? No. Does it create any variation? No. They are absolute clones. Which animals are clones? We've said that. Why are they clones? Blah blah blah. They've got the same DNA, they've got the same genes, the genes are the code for the characteristics. At which point does asexual reproduction occur? Well, I suppose here you take the nucleus out. There, at this point, this is going to be a clone of that. Okay. So these two that are tricky, let's just flip back and forth again. We've got fusion cell cloning, and now we've got embryo cloning here. And this one, there's a lot going on here. If you see you've got a mother here, you've got a father here, you notice the father hasn't got any udders. This is supposed to be a sperm, here's an egg. Well, this is sexual reproduction here. Even though the whole process involves cloning, you've got sexual reproduction here, sperm and an egg, just like normal sexual reproduction, and as you know, sexual reproduction creates variation. So the next generation are not clones of the parents. They will share the characteristics. They'll probably have roughly half the characteristics of the mother and the father, but they are not clones of the parents. So we've got sexual reproduction here creating variation. We've got an embryo, and at this point the embryo is split. So this is the point of cloning. Cloning happens here. Now all of these cells have identical nuclei, and again the nuclei contain the chromosomes, and the chromosomes contain the genes and the DNA, and the DNA is the code for characteristics. It's the same blurb, it's the same keywords I keep coming out with. So at this point you can see again, this develops further into an embryo, is implanted into surrogates, 3 different surrogate mothers, not related at all to anything else on the screen. And then as soon as the embryo is developed, you've got 3 clones born here. So what are the clones? These are clones of each other because they've all come from the same embryo, and every cell in that embryo has the same DNA, has the same genes. So that's confusing a little bit because you've got sexual reproduction happening here, and here you've got asexual reproduction. That's the point of cloning. Right, so now these two you don't really, it's not specified that you need to learn these from AQA, but they're quite interesting anyway. One of them is inbreeding, one of them is crossbreeding. For inbreeding, you select two animals that have similar characteristics. So in the hope that you're going to develop those characteristics, maybe accentuate those characteristics even more. So this one is inbreeding, you can see is sexual reproduction. Yes, sexual reproduction does cause variation, but because you're choosing two parents with such similar characteristics, in all likelihood, probably you'll select, you'll create an offspring with lots and lots of milk. Now sometimes there can be problems with this. If you select a characteristic that's on a chromosome that's got other problems associated with it, you can cause problems in the offspring. A common example is breathing in dogs that have problems breathing. So that's crossbreeding, sorry, that's inbreeding. Here you've got crossbreeding, two different characteristics. You can see you've got a male, you've got a female, and you're crossing them in the hope that you're going to get a good combination of characteristics in the next generation. So we've done quite a lot there, and those diagrams look absolutely bewildering, but you should be able to look at any one of those diagrams and identify, point at the screen and say, yes, that's adult cell cloning, that's or fusion cell cloning, that's embryo cloning. Oh, I've just noticed that diagram's a little bit different to the one I showed you earlier, but you can see that's embryo cloning because the embryo's split. Okay, that table I showed you earlier, if you like summary tables to compare the differences, there's one I've completed there in a bit of a hurry, but is a rough summary. Okay, good luck.